All right. So today we're going to be talking about some basic principles of how you model regulatory and signaling networks. And it's going to be a little bit of background information. We actually used some of this last week, although we hadn't really explained the details. And so we're going to be talking about Michaelis-Menten and Hill rate laws. Again, if we were doing a mathematical biology or a computational biology class of a classic type, we probably would spend a week or two on this. And there are additional slides, plenty of additional slides in the deck that I'm going to skip over today, but you can refer to. The textbook also has quite a bit of additional detail on this. Now I'm going to try at the bottom of on the bottom of the slides, I'm going to try to remind people what parts of the textbook I think are going to be most useful for people to review. However, I also encourage you to look at the textbook and see what's appropriate. And also, if you don't find that the textbook has the material that you need, reference other sources. I've pointed you to that online textbook from Elowitz, and I asked in the homework for people to look at least one other textbook. And the reason for that is that this is still a fairly young field and it's a fairly diverse field. And so the things that one person finds clear and helpful may be too detailed or too confusing for somebody else. And so, again, I find Herbert's textbook particularly simple and clear. It doesn't cover as much material as some others, but there may be other resources that you find are more suited to your particular background and interests. And as always, I've been putting these up online on YouTube. I understand that the unedited versions can be a bit of a slog to look at, and it takes me sometimes a few days to get them edited, but I do want to make them available to people. So I'm going to start out by reviewing a couple of concepts that we went over in the past week, and also perhaps a few things that were more or less random points that we haven't discussed, although they're put in more context in the textbook. And then I'd like each of you to tell me a little bit about that homework assignment where I was asking people to begin to think about possible project ideas. Don't have to be committed to anything at this point, but I'd like you to present 3 or 5 minutes about your ideas from the homework. And then we'll move on to some exercises where we'll define a simple enzymatic reaction. We'll do a little bit of computing on that. And then we will introduce the concept of a Hill function. And depending on the amount of time, I would like to show you how to download models from the web and run them and also play with that a little bit. And that's a sort of an open-ended exercise. If we don't do it in class, I may ask you to come and do that on your own. Herbert wrote a nice package that allows you to run antimony models online, not in a notebook the way we've been doing it, but in a browser interface. And some people may find that interesting. One of the things that it does allow you to do is allows you to translate models from antimony to system biology markup language, SBML, into some other formats as well. And that's quite convenient. So there's one tiny bit of notation that I want to get out of the way, which is we've talked about one of the fundamental concepts in especially signaling and also in gene regulation is the idea of a complex where you have two molecules that come together and that do something, and that is usually written as a chemical transformation. E plus S goes to ES. And if you're talking about the names of chemical species, then that's unambiguous. But if you're talking about the concentrations of the species, which are the italicized versions of those names, E next to S usually means E times S. And so it's ambiguous when you write ES in italics, whether you're talking about the concentration of the species ES or the product of the concentration of E and the concentration of S. And so in my world, I will try to remember always to put a dot so that if it's E times S, I will write E dot S. And if it's ES, it's the amount of the complex ES. Other people like to use brackets, square brackets, to indicate the concentrations of a species. Sometimes people will use lowercase italics for the names of the species. Sorry, for the concentrations of the species. And so I'm just giving you a heads up that there's some inconsistency in the notation. And when you're doing enzyme reactions in particular, you're likely to find things that could be confusing. So be aware of that. The second thing, which we've already talked about, are these dangling arrows where we don't have something on the end of the arrow. Nothing goes to S or S goes to nothing. And in general, when we do want to put something on the end of those arrows, we have to think about whether that is a floating species whose concentration will change, or what's called a boundary species, which, for example, could be supplied from a reservoir where that concentration doesn't change. And so my example here is nothing goes to S at a rate K. That's a constant source of S. I can also say, well, in fact, that is supplied by molecule A, and it's supplied at a rate K. And if I do that, A will go negative unless I assert that A is somehow magically held to a constant value. And that makes A what's called a boundary species. And so in antimony, the dollar sign, I'm not quite sure why Herbert picked a dollar sign for this, but the dollar sign locks the value of a chemical species to its initial value. And so if you want to explore, and we'll do this a little bit later, what happens when a species floats versus when it's constant, you can put the dollar sign in, and that will lock the value of the species. In antimony, if you put a dollar sign in front of a species name once anywhere, that locks the value everywhere. And so if A is used in seven different chemical reactions, if you put a dollar sign in front of A anywhere in that system, that will lock the value. We've already seen first order decay, S goes to nothing, and that's usually at a rate proportional to the amount of S. If we said S goes to something else, then that something else will typically accumulate, and that can make plotting things difficult. And so we may actually also lock the amount of a target as well. Another little thing to worry about or think about when you're doing dangling arrows is that the stoichiometry is ignored when you do a dangling arrow. So if I write nothing goes to 3A at a rate K1, that's interpreted as nothing goes to A at a rate K1, not nothing goes to 3A at a rate K1. You don't multiply by 3. You do in every other kind of stoichiometry, but in that particular stoichiometry, you don't. Similarly, if you have 2B goes to nothing at a rate K2 times B, the rate of decay of B is not 2K2B or half K2B, it is K2B. And so that's a bit of a notational inconsistency that you have to be aware of. Your values may be off by the stoichiometric coefficient if you're not aware. Okay. And so this is just, again, the interpretation. Nothing goes to 3A, 3A goes to 2B, 2B comes back to 3A, 2B goes to nothing. You have to split that up into three separate reactions. Nothing goes to A, 3A goes reversibly to 2B, and B goes irreversibly to nothing. Again, when we do enzymatic reactions, you'll see that show up. Last time we overwrote antimony values from Python, and we also learned how to use resets so that we could do parameter scans. And I just want to remind you of how to do that. You'll have those in the notebooks that you've been developing from previous classes. And I strongly encourage you always at the beginning of class to open the notebook that you finished with from the last class, launch the installer so it gets through its minute of installation, and then you can do that everything in the class in one notebook. Okay, we talked about using plot array to plot multiple time series in the same plot. You can certainly use matplotlib functions if you're more comfortable with those. Within plot array, there's this auto color cycle, which has the sort of perverse name of reset color cycle equals false. If I were naming it, I might have called it color cycle equals true instead of the opposite, but anyway. And how to label or give a key for that as well. And again, we did that last week, and you'll have that available. We talked a little bit about chemical equilibria, and we're going to be distinguishing a little bit the difference between a chemical equilibrium and a steady state. A chemical equilibrium only applies to a system that's a closed system. So in this example, I have A goes to B, B goes back to A. I've written a simple mass action rate law. The rate A goes to B is Kf times A. The rate B goes to A is Kr times B. And if I allow that system to evolve in time, A and B will reach constant values, where the ratio of B to A is called the equilibrium constant, K equilibrium, and that's going to be a Kf divided by Kr. Notice that while the ratio of B star or B equilibrium to A equilibrium is constant and independent of the initial values, the total amounts of B and A are going to depend on what the initial values were. The sum of A plus B is conserved in this reaction. When you're talking about chemical equilibrium, this is why people like them. That's why you do things like flux balance analysis. Is that even for very complex chemical reactions, if I had a many-component chemical reaction with complex stoichiometry, K equilibrium is basically at least for mass action here, is the concentration of each component raised to its stoichiometric coefficient on the left divided by the right. And that's it. And that's just, and the equilibrium amount is just Kf over Kr. And so the system behaves as if there were only a single component on either side. And that means that in particular, if I do a chemical reaction where I mix two things in a test tube and then measure the amounts after they've reacted, I can get K equilibrium very easily. And so from an experimental point of view, measuring K equilibrium is quite easy. Measuring the lowercase Kf and Kr is in general quite difficult. However, in living systems, we're almost never in a closed system. We almost always have an input of something and an output of something. As they say, the only real equilibrium is being dead, and even that's not really an equilibrium because you're decaying. And so here I have a system that looks just like my previous system. A goes to B, B goes back to A with exactly the same rates, but I have a constant source of A, K1, and I have a constant removal of B with K2B. And this will also reach a steady state where the values of A and B won't change in time. And that's called a steady state rather than equilibrium because I'm continually adding material in the form of the source of A and removing it in the decay of B. And I have a bunch of slides doing the mathematics, which I'm going to skip here, but it's worth looking at what the final values of A and B are in these two cases. They're reasonably parallel to each other, but they're not the same. In the case of the open system, A star over B star is equal to K reverse over KF, but you have K reverse plus the decay rate K2. In the closed system, the equilibrium ratio is KR over KF. So there's this extra K2 that shows up in the ratio. Notice that the ratio A star over B star in the open system does not depend on the rate at which I add material. It's independent of K1. The equilibrium concentrations, A star and B star is actually pretty simple. B star is just K1 over K2, the rate at which I put material into the system divided by the rate at which I take it out. What's interesting about the value of B star is that it's independent of the forward and backward rate constants, Kf and Kr. And that might be a bit unexpected. You might expect that if the forward rate Kf were bigger, a B star would be bigger. It's not. A star does depend on all of those things. You'll notice that A star and B star do not depend on the initial values of A and B. They're independent of the initial conditions. If I look in the closed system, on the other hand, the A equilibrium depends on the initial amount of A and B, and the same for B equilibrium. And both of those depend on Kf and Kr. And so the ratio of the two equilibrium amounts is parallel, although not identical, but the amounts themselves in one case are independent of the initial condition, in the other case dependent on it. In one case, they depend on the source rate. In the other case, of course, there is no source rate. And so even though these systems are similar in some ways, their dependence on the parameters is quite different. And if you're interested in the mathematics of that, any mathematical biology or chemistry textbook will have chapters on all the analysis. This is the simplest possible example, but they'll show you how to analyze very complex systems and get those equilibrium or steady states. I mean, one thing that comes up from the point of view of trying to build models and validate models is that classically, gene knockouts were the easiest thing to do. You got rid of a gene entirely. But if you actually want to build and validate the kinds, at least especially the dynamic models that we tend to focus on in this class, what you really want is to be able to modulate the level of a gene with essentially the smallest perturbation that has some measurable effect. Because you actually want to be able to calibrate. If I change the strength of this signal here, how does the system respond? And doing a full knockout, as Ellen points out, is often lethal. And in that case, you can't make any downstream measurements except lethality of it. You can't say how do the chemical concentrations change because you killed the cell. And so if you can turn down a gene without being lethal, then that gives you a lot more control over making the measurements of the effect of that gene. I remember when we were doing chemical toxicity experiments, they thought, well, the easy thing to do would be to give things arsenic because we know it kills cells. Well, that's actually a very hard thing to analyze because if all you do is you have your dish, you put your chemical in the dish and all the cells die, you've learned nothing at all. What you need are actually graded response experiments. Now, that doesn't mean that knockout experiments aren't interesting, but they give you somewhat more limited information than graded response experiments. But Ellen here has spent 20 years of her life working in bacterial systems. And so how many? 30 years of her life working in bacterial systems. And so you definitely please brainstorm with her about possibilities.
When we're doing like dynamical models and like in this class, is that pretty much all going to be some sort of regulatory thing more so than like this one?
Oh, people definitely do dynamic models of metabolism. I mean, the reason historically that people did flux balance analysis for metabolism was twofold. One, the models were huge, and so computationally it was expensive to do dynamic simulations. That's less of an issue today. And two, exactly what I showed in those first slides of the class, which is that people could measure the equilibrium constants of reactions, but they didn't have the ability to independently measure the forward and backward rate constants. And so for flux balance analysis, you just need the equilibrium K. You don't need the forward and backward rate separately. Precisely because of what I said, which is the output doesn't care about what's happening in the middle. And so I put those slides, I actually taken those slides out of the lecture, and I put them back in precisely because of your interest in. They may have seemed a little bit off topic, but they were there because of your thought of your interest in the FBA type things. And so I'm trying to think who's I mean, there certainly are big textbooks on metabolic modeling, which focus on flux balance analysis. And I'm trying to think of the standard one now. I'm not going to pull it up. I have the book on my shelf, and I can even visualize what the cover looks like, and I'm not pulling up the Voigt. Eberhard Voigt would be the classic one. And so you might look in Voigt and sort of and try to think about what kinds of questions. Look at the homework problems in Voigt and say, okay, now starting from the kinds of things he's assigning as a problem to do, how could I generalize that to make it something that's interesting? Okay, great. Well, we've had a longer discussion than I planned, but you've also thought in a very sophisticated way about these things. So I'd much rather take time to talk. The basics that I'm going to be covering are things that I'm happy to do with you, but also are things that at some level probably you can learn independently as well. Any other comments or suggestions before we move on? People have an idea for. I'm not going to have a formal assignment for next week, but I probably should do that. Whereas I really do want people first try to set up an appointment with me or Pedro or both, or maybe you with Ellen. And then I think we do need to have a weekly schedule, and I didn't put that into the homework assignment, on trying to make weekly progress on this. And so given these ideas today, I think the critical thing for next week, and I can add that to the homework, will be to try to move forward. And you've got three wonderful ideas or four wonderful ideas about possible projects. Let's try to work for next week on finding, maybe Pedro, you can make a note of this so we can type it up. First, finding some papers where there are published models that are relevant. And the second thing, and then look at those papers. And then the second thing would be to try to narrow down a little bit what would questions that could be answered with a model look like. And so if people can work on that for next week, then I think we can move forward and begin to turn these great ideas into actionable projects. Okay. the things that I'm going to be covering today, we're not going to have as much time, not that much time to cover it, but the things I'm going to be covering today are quite elementary. But at some level, we have to get the elementary stuff under our belt to be able to do these ambitious things. And so I hope it doesn't feel like, I think the expression, I, I don't know, maybe it shows my age, but are people familiar with the website TV Tropes? No. None of you are, you're all too serious to do things like watch television. None of you watch television. There is an enormous wiki on the conventions that show up primarily in Japanese anime. It's mostly about anime, although it also has live action movies and TVs too. And it gives these very detailed formal definitions of all of the structures, the conceptual structures that exist in storytelling. It's enormous. I mean, it's almost the size of Wikipedia, this thing, contributed by fans. And so in any case, occasionally you'll hear me reference TV Tropes because it has the best definition of some concept anywhere. Now everybody will go start reading TV Tropes. It's an unbelievable time sink. So I'm not sure I'm recommending it to people, but in any case, if anybody is ever interested in the not very fashionable subject, which I'm interested in, and Ellen is, of ontologies, how do you formalize our way of talking about things? There is no better source for ontologies to learn about how to build ontologies than TV Tropes. It is the best example of a large-scale formal ontology anywhere. It's much bigger than any of the medical ones. And it actually is more careful. I don't know, but I don't think probably our students this year are that interested in ontologies. You might be because of the things you're working. How do you actually formally describe what you're seeing in your histology slides? Those are so do you use mesh or Go or other ontologies to annotate your, okay. So from my group, Jim Sluka doesn't come to these classes, but he's an ontologist. So I actually have people in my group who spend their time worrying about that. All right. And it's tricky because if you don't have standardized terminology, then things get very, very confusing. How do you look things up if the same gene has 10 different names? It's not easy. All right. So now we're going to do, after thinking about these grand visions and big ideas, we're going to come back to some very, very simple models in computing. And I want to thank Pedro for providing this slide for us for today. Pedro roughed this slide out this morning for you. Because what we're going to be talking about for the rest of the class today, and we probably won't finish it, but we'll see how far we get, is a very, very simple concept, which is, suppose you start out with some signaling or regulatory diagram, and you're not, essentially, you're never going to have completely detailed information about the dose response for the components of that diagram. And so there is a standard assumption that, and I should have probably put a sigmoid on this plot so to explain what a Hill function looks like. There's a standard assumption that the dose response of any kind of signaling or regulatory network is a sigmoid. If you have an activator and you have very little of the activator, then you produce a low amount of whatever that product of that reaction is. As the amount of activator increases, the production goes up very slowly at first, and there's a region in which the rate of production goes up fast, and then that amount will saturate. And so you get a sigmoidal response. And that sigmoidal response is typically modeled numerically as what's called a Hill function, and we'll show you the mathematics of a Hill function, which is very simple. And as this slide points out, well, we often use Hill functions essentially to encapsulate our ignorance and to encapsulate that ignorance with the minimal possible number of parameters. Hill functions actually do result from enzymatic reactions. And in the class, I don't know if we'll have time to get to that or not. But if we do the mathematics, we can actually derive the Hill function for a multi-stage enzymatic reaction, at least for the steady state. So Hill functions for multi-stage enzymatic reactions give you the correct steady states, but the kinetics, the transients are a little bit off. And so we're going to do a couple of steps that are going to get us to that derivation. And there's a chapter in Herbert about it and elsewhere as well. Sort of a classic example. But when you're talking about regulators, either positive or negative regulators in particular, the classic way of doing that is through Hill functions. And so the first thing that we have to do is introduce the idea of an enzymatic reaction. And so here we have an enzyme E, and that enzyme is going to complex with S to form the complex ES. And then E and S will react to release E. Enzyme is not used up in the reaction, but we wind up with a product P. So fundamentally, we have the chemical reaction S goes to P, but the rate of that reaction is controlled by the amount of enzyme. Something that we have to remember here, if we want to solve this thing analytically, is that the total amount of enzyme is conserved. The enzyme shows up in two places here as free enzyme E and in ES. So E plus ES is a constant, which is the total amount of enzyme. And that has the fancy name of a conserved moiety. And it means that when you do things like flux balance analysis, if you don't get rid of that, the matrix is singular. You can't invert it. And so in the numeric solvers that were implicit solvers that Herbert used to use, if you didn't tell it explicitly to look out for conserved moieties, it would diverge. These days, that's turned on automatically, but you may see some of my older code where it says moiety conservation equals true to make sure that you use the solvers that check for these kinds of divergences. And this is why I said at the very beginning, I will use E times S to denote the concentration of E times the concentration of S, and ES to denote the amount of the complex, the concentration of the complex. As I mentioned last week, when you run r.simulate, it gives you a table where each column of the table is labeled with the name of the variable. The first column is time, and then each of the other variables in the problem is another column. You can select what columns to output in your simulate function with a list, square bracket, a name of variable, comma, name of variable, comma, name of variable. That can be convenient if you have very complex models. Then you don't need to be digging through hundreds of different columns. You only put out the ones that you want. You have to remember that time is a special reserved word. It's quote time with a capital T. The other thing is that when we've done these simulations, you've named the rate laws, J colon or R colon something. And if you do quote R1, that will output that rate, which can be useful too. So the first exercise, and we'll see how far we get today, is to write this little enzymatic reaction. This is to test our remembering how things are going. So write this little enzymatic reaction. Start out with E0 equal to 1, S0 equal to 10. ES 0 equals P0 equals 0, no product and no complex. And you can set all the rates, the forward and backward rate and the production rate to 10, sorry, to 1 to start with. And so why don't people fire up to antimony and try implementing that? And I realize that this is pretty simple compared to the ambitious things that we were talking about a few minutes ago. But those ambitious things that we were talking about a few minutes ago are going to be composed of hundreds and hundreds and hundreds of these. So let's get one of them working on your computer first. One thing to think about if you're trying to analyze something like this and turn it into antimony is in antimony, every time you see an arrow, you're going to have one reaction. So here there's a forward arrow, a backward arrow, and a product arrow. So that means you're going to have 3 lines of antimony specification.
So the top one is Kf times concentration of E times concentration of E.
Right. And the back reaction is Kr times the concentration of Es, the product. So these are both mass action laws. And this is precisely why I had that slightly obscure comment about the notation at the very beginning of the class. And you should wind up with something that looks more or less like the plot on the bottom right. And I've done something a little bit tricky here, which is in the simulation I ran, I only ran it for a short amount of time because I wanted to illustrate something. But if you've got it working, run it for a longer time and then tell me what happens. If you've got it working, I would encourage you to run it for longer and see what happens, because as I say, I've selected the early time behavior for a particular reason, but the long time behavior is actually rather different from what you're going to see here. And the other thing that you could do, which could be interesting for you, would be to change the changing the amount of enzyme doesn't do anything very interesting to this. But if you change the value of R with respect to Kf and Kr, so if you make R much smaller, much bigger than Kf equals Kr, that will give you different behaviors. And if you make Kf not equal to Kr, that will also give you different behaviors. So a lot of the time when I give exercises like this, if you've got the exercise done ahead of the other people in the room, try exploring what you can do with it. Now what you'll see is that S here is decreasing and P is going up. And so if you only plot ES, well, ES and P isn't a great choice, but if you plot S and ES, then you'll see the blue and the green lines in more detail. Okay, so this exercise was just in column selection. If we plot just ES and P, you should see these two. If you wanted to plot the rate of production of P, then if you named, for example, your reaction J3 colon ES goes to E plus P, and you do M equals R dot simulate time, comma, quote J3, then you'll just see the rate. So why don't people try plotting that instead? Of course, if you named that reaction something else, you'll have to use whatever name you used instead of J3. So what you should see is something that looks like this, which is that the rate of production of the product goes up very, very fast at the beginning, and then it gradually tapers off as the amount of substrate goes down. So now we're going to use that boundary species convention, which is we want to compare what happens when we allow the amount of substrate to gradually decrease to what happens if we hold the amount of substrate fixed. And to hold the amount of substrate fixed, in your simulation, you put a $ in front of the S somewhere. Find one of your S's, make it $S, and run it again. And since we don't have too much time, I'm not going to ask you to overlay the plots for with and without the dollar sign, although if you could figure out how to do that in a quick way, by all means do it. So this exercise consists of doing exactly one thing, which is adding $1 sign to the simulation and running it again. Now, in the online version that I have, I actually did write it so it overlays with and without the S. I did that in a pretty dumb way. I simply copied the simulation code into duplicate the simulation code, and defined one antimony model with the dollar sign, one antimony without, ran it once with show equals false, and then ran it a second time with the other one with show equals true. You could be more elegant than that. But if you look at the result, what you'll see is that in this period from time 0 to time 5, the results are not too different from each other. But as I get close to time 5, the amount of product that I have when S is not held constant is a little bit less than the one that I have when S is held constant. And this makes sense. As I'm running the simulation, S, the substrate, is being used up. And so as the substrate is used up, the rate of production will go down. And if I run this for a long time, like 100, I'll run out of substrate entirely. And so in one case, I'll keep producing sub-product. In the other case, the rate of production of product will go to 0. Can people see the difference between those two? Does that make sense? The next thing to do is the derivation of the Michaelis law, which is now you want to scan the rate of production, which you've just plotted, versus the amount of enzyme. And one way you can do that, and maybe we can just go through this quickly. There are a lot of different ways you can overlay plots. One way we've done overlaying plots was to use the show equals false and then show equals true. Another way to do it is actually to assemble all of your different time series into a big array. And I'm not going to have you do this. I'll just walk you through it. You can look it up. This is the kind of thing these days which probably you look up in LLM instead of remembering it. So one thing you can do is you can run the simulation once and just get the time vector. And then you can run the simulation again and just ask for the rate. And the reason you do it this way is because if you're going to combine arrays, the number of rows in the array have to match. Otherwise, it will crash. You can't combine them. And so you do have to be a little bit careful to make sure that you have everything in alignment. That's not so much of a problem when you're doing these deterministic ODE-like models. If you're doing stochastic models, that turns into a problem. There are ways around that, but we'll get to that in a week or so. So you can use a function like numpy hstack, which combines arrays together horizontally to assemble things. And for today, I don't want to spend time on that. I'd like to get more to the science. But if you want to use a function like numpy hstack, last week we used append for lists instead of doing it this way. Append is a more Pythonic way of doing this. Hstack is a more procedural way of doing it. Hstack takes two arrays and glues them together side by side. And if the number of rows in the array don't match, it'll crash. And so if I do a simulation like this, M equals R dot simulate 0, 20, 100, and then I select the time column. That gives me my x-axis. And now I go for S in some numpy A range. That's the parameter I'm going to scan. I reset my simulation. I set the value of S. I now do N equals R dot simulate 0, 20, 100 again. And here I plot the rate of production J3 versus time. And now I H stack M and N together and output that as M. And each time I run this, it'll add one column to my array. So can people try that out? If basically you have to add this line M equals R dot simulate to get the time, for S in N pi A range, reset R dot S equals S, simulate M equals N pi H stack, and then you can use a plot array to display the results. There are plenty of other ways you could accomplish this. As I say, you could use plot array with show equals false and not assemble the data by beforehand as well. But if you wanted to post-process the data sets, you probably have to do this kind of assembly. Yeah, compare it with and without the dollar sign. You're allowed to have it more than once, but if you have it in front of any occurrence of S, it will freeze S. All right, so you should have gotten something like this, and I apologize for the code having disappeared when I launched PowerPoint this time. This is without the dollar sign. Now what you see is that the rate of production goes up and then decays as the substrate is used up. All right, and we've already talked about how to do labels and lists and things. I'm not going to talk about that now. Okay, so now what we want to do is plot the final state versus S. And this only works for the dollar sign because the final state, without holding S constant, you run out of substrate. So the final state is 0. And so what we want to plot is the rate of production of P as a function of S when S is held constant. And originally I used the get steady state function in Tellurium, but we're not going to do that. We're just going to run the simulation. And so here I'm not going to use the VStack. I'm going to use the append version of this. You have this code from last week. So you'll say L equals empty list. So this should say for S in N pi A range, reset R dot S equals S, simulate append, this should be S, R dot, and then instead of R dot A, well, here's the VStack version of it. I was trying to avoid using VStack, and so, but I pasted in the code from last week. I forgot to paste in the corrected code. Structurally, it's correct. The key thing is we want to, well, maybe let me just do it.
We're just trying to get like the last value.
Right, you want the last value of J3, the rate of production of P, and you want to have S versus J3 as a plot. So you want to come up with this picture. And here is code that will do it, but it's using the VStack command rather than the append command. The VStack, the append is a little bit simpler to type. And here I actually used get steady state values rather than just running the thing for a long time. You should not need the conserved moiety analysis equals true. That's old code. And the reason that the append version of this is a little more convenient than the VStack version is that when you do a VStack, you have to create an empty array to start with. So you need that NP empty shape equals 0, 2, which is always hard to remember, at least for me. But if you type exactly the code that I have here, it should work. Or if you remember the code from last week of using append, that'll work too. In any case, you want to scan S from a value of 0 to 10, and you want to get the long time value of J3 for that value of S and plot. In particular, this is telling you how the rate of production of product depends on the amount of substrate. And in the slide deck and in the textbook, there's an analytic derivation of the Michaelis-Menten law, which is what this is. And actually, my online notebook has both kinds of code. If it's not smooth, then you should change the A range step size.
Yeah.
And I don't know why I started with S at 0.1. I should really put it near to 0 to begin with. If you don't want to use the get steady state values, you can just run. Oh, so before you do a plot array, you have to turn the L into a NumPy array. I did not do the derivation, but for frozen values of S, you'll find that at steady state, the amount of complex is the total amount of E times S divided by this somewhat complex looking thing, KR plus R over KF plus S. And if I define the Michaelis constant to be KR plus R over KF, then the amount of ES is equal to the total amount of E times the S divided by Km plus S. And then I can further simplify by defining V max, which is equal to R times E total. And I wind up with the Michaelis relation, which is that the rate of production of P is equal to V max times the amount of substrate divided by Km plus the amount of substrate. And that's the plot we were trying to generate just now. And so this is actually now a derivation of that. And you can also write this in what's called a dimensionless or unitless form, where you divide S through by Km. So you scale S relative to Km, the Michaelis constant. I haven't changed anything. I've just multiplied and divided by Km. And this tells us that if S over Km is much less than 1, then the rate of production is basically V max over Km times S. And if S over Km is much bigger than 1, the rate of production is V max. So for small concentrations, small relative to Km, the rate of production is a linearly increasing function of S. And for large concentrations compared to Km, the rate of production is a constant. Now, in real experiments, that actually is validated pretty well, but it only applies at steady state. And so there are rules about when it applies or not. When the amount of substrate is much bigger than the amount of enzyme, it works. So you're enzyme limited rather than substrate limited. And when the rate of production R is much less than KF and KR, then it works. It will fail if those conditions fail. And so now we're going to do a slightly more elaborate example, and this is as far as we'll get, I think, today. which is now suppose that I had either multiple substrates being used simultaneously. So I have KF, so I have E plus N units of S making a complex and making product. Or I imagine a stacked series of reactions. E plus S goes to ES, ES plus S goes to ES, S plus ESS plus S goes to ESSS, and so on. Well, that's a cascade reaction. In reality, you almost never have, it's hard to imagine how you could have 10 units of S bind simultaneously. So the second one is actually probably more realistic. And the question is, what is the rate of production of P as a function of S? And so to do this, you have to take the code that you just wrote, and you have to change precisely one line of your code, which is instead of E plus S goes to ES at a rate KF E times S, you have E plus NS goes to KF E times S to the N. And remember that in antimony, unlike in Python, the exponentiation operator is the caret. And if you don't want to use N as a variable to begin with, you could just make N equals 4. That's a good choice. And so you could say E plus 4S goes to ES at a rate KF times E times S to the 4. And what did you get? So you're only changing one line of your code. So all you're doing is changing the line where you had the reaction from E plus S goes to ES to E plus 4S goes to ES, and the rate from KF times E times S to KF times E times S caret 4. So did somebody get something that looks like that? This curve for N equals 4 is somewhat different from the Michaelis curve. For very large values of S, it saturates at V max, just like the Michaelis curve. At 0, it's 0, just like the Michaelis curve. But in the middle, the Michaelis curve has the maximum slope at 0. This has a maximum slope at K, at Km. And the slope is actually 0 at 0. So the only line you changed was this J1 line here. The Hill function has the form E times S to the N over, and I always raise K to the N, so everything is symmetric, K to the N plus S to the N. The dimensionless form, you divide S by K. S is also known as the half maximal concentration because this function is equal to 1/2 when S equals K. And K is known as the Hill constant. And so the final result is that the rate of production of P is V max S to the N over K to the N plus S to the N. And that is a very fundamental concept, the Hill equation. Because when we do regulation, we almost always use the Hill approximation for regulatory interactions. We're going to run out of time today, but I want to go through this concept because you'll be using it in the homework, and we'll come back to it next time and apply it, which is, suppose that we see a regulatory arrow. A promotes S2. Or a diagram where we say S1 goes to S2 and that reaction is promoted by A. That would be in a signaling network. suppose that we don't know anything else about the system except that diagram. Our first thought might be, well, if we just have the diagram and we don't know anything else, there's no way we can convert that into a computable model. And at some level, of course, we're right. If we don't know anything, we can't solve it. But there is a way of constraining our ignorance. So if we see that A promotes S2, and we don't know anything else, we assume that that means that nothing goes to S2, and the rate of production of S2 is regulated by A. And because it's a lollipop arrow, that means it's an activator. So that means that if we have no A, we don't get S2. If we have a lot of A, we get some S2. We also know that in biology, we never have infinite rates. There's always some maximal rate of production of anything. And so when we see that diagram by itself, we automatically interpret that as Vmax times A to the n over K to the n plus A to the n, a sigmoid, the Hill function. And there's a great thing about Hill functions. Hill functions have three parameters, Vmax, Kn, or K, and n. N is the steepness of that sigmoid. If n is bigger, the sigmoid is steeper. And we'll discover that for most cases, the value of n isn't that important. If we pick a value of n of equal to 4, 6, 8, 10, we get more or less the same result. If we make n less than 4, if we make n equal to 1, we get the Michaelis function, which is actually quite different. But for n between 2 and 8 or 10, basically the value of n doesn't matter that much, at least to first order. Kn is something that scales the concentration. It's the half maximal value. What we'll typically do if we don't know anything is pick Kn to be the same for every molecular species that we have. And that means that the input and output concentrations of each species are compatible with each other. And that leaves us only with one parameter, which is Vmax, which is the speed of the reaction. It doesn't change the amount, only the speed. So the question was, why is setting K equal to the same value everywhere a good idea? If we use Kn equal to a half, then if A is less than a half, we basically don't get any reaction. If K is bigger than a half, we get a reaction. And suppose Vmax was always set to 1. Then A to the n over K to the n plus A to the n is a number always between 0 and 1. And it will have a value of a half when if Kn is equal to a half. And so in that case, everything will be set up so that the concentrations always scan between 0 and 1. And so the output of one circuit will always be compatible with the input of another. It's a little bit like trying to reduce this to a logic gate. In TTL logic, you use 0 volts for 0 and 5 volts for 1. Here we're trying to say a large value is A equals 1, a small value is A equals 0. And as I think I mentioned last week when we were doing the autocatalytic circuit, Herbert set capital K equal to 10. And that scales A between 0 and 20. And I made the sort of cryptic comment that I like to use capital K equal to 0.5 because that keeps the value of A between 0 and 1. Okay.
It is arbitrary, but the reason why we can be arbitrary regarding the Hill functions is that most of the time you don't have access to the actual measurement of the protein concentration inside the cell, like an enzyme, like the concentration of the enzyme inside the cell. Like it's rarely you'll find the exact numbers in literature. So you basically choose the units you prefer.
Right.
And in our case, it's 0.5 for K. And your protein will be between 0 and 1. And if it happens that you find the actual concentration of the molecule in real life, you just say 0.5 means whatever moles per milliliter
Yeah.
of the real measurement. So you just rescale things, and then you're good to go. So yeah, that's what.
Remember that most biological experiments report fold changes. When I double the value of, when I double the amount of S, the amount of A doubles. Right? You get, most biological experiments give you fold change results. They don't give you absolute concentrations. Because typically you're measuring, well, if you're lucky, you're measuring fluorescence intensity, which actually gives you the amount of protein. A lot of the time, what you have is the amount of RNA, and you don't even know how that translates into the amount of protein very well. Your hope is that if you have more of the RNA, you get more of the protein, but you don't know what the conversion factor is. And so it's almost never true that you know the absolute amounts of things. You very often do know that the amount doubled, but you don't know what the reference amount is. And so that means that rescaling things is something that biologists do absolutely all the time when they do experiments. You don't know the absolute scale. You know that there's twice as much virus as there was in the other sample, but you don't actually know how much there was in either sample. And so those are good questions to ask. And of course, if you can actually make experimental quantitative measurements, the exact amount, that's wonderful. But most biological experiments don't allow you to do that. I mean, there certainly are some. I mean, there are experiments where you actually count individual messenger RNAs. You label each one with a quantum dot and you say there's 1, there's 2, there's 3, there's 4. But with proteins, typically you don't know that. Even if you're doing things like mass spec, you're getting relative amounts. The other thing that's worth mentioning is that there's something very symmetrical about the Hill equation. And I've skipped a little bit the bottom one, but I can do that here. In the top one, we have R inhibits S2. So that's interpreted as nothing goes to S2. The more R we have, the slower our rate of production of S2 is. The beautiful thing about the Hill equation is that the only thing I have to do to make the Hill equation into an inhibitory circuit is to take the variable at the top and replace it by K. So let's look at that. Here I had V max A to the N over K to the N plus A to the N. All I do is swap out the A with K, and I've called it R for inhibit. It's typical. R is A stands for activator. I'm not sure why R stands for inhibitor, but that's the standard notation. And so if I want to say what happens if this is an activator, I just change one letter in my code. I don't have to do anything fancy. And because I've scaled everything with the same K, everything stays compatible. And that's actually makes things really easy to do. It makes it very easy to build circuits when you do things this way.
You only do that for like signaling, or if you're.
Well, so this will work for signaling and gene regulatory networks. For metabolic networks, in general, we're having to worry about actual chemical transformation. And the bottom one here is thinking about a chemical transformation that's an enzyme. So this would be more like a metabolic network. S1 is becoming S2, and the rate of production of S2 is inhibited by the presence of an enzyme R. That's possible. Usually we think of enzymes as activating, but let's just, for purposes of the discussion, let's assume it's an inhibitory enzyme. Okay.
So you can't, in that case, you can't set K is the same for all of them because you can't, it wouldn't be like. You can't treat all of them the same, but more based on the production constant as opposed to like.
So here you break apart the problem into two separate things, and that's what these two columns here are about. I have one question, which is, suppose that I didn't have any regulation at all. What is the rate at which S2 is produced by S1 in the absence of any regulation? Suppose that S1, and it's even written correctly on the slide. If S1 is assumed to become S2 according to mass action, the rate of production of S2 is proportionally that of S1 that I have. Then, if I had no regulation, the rate of production of S2 would be Vmax times S1. That would be it. In this case, it's regulated by R, so I multiply by K to the N over K to the N plus R to the N. Remember, K to the N over K to the N plus R to the N is a number between 0 and 1. So increasing the amount of R decreases the rate of production. So that's what I want. As I increase the amount of R, I get less. If I do the activator version, it looks exactly the same. The unregulated reaction is Vmax S1 times A to the N over K to the N plus A to the N. If I have no A, I don't produce any S2. If I have a lot of A, I produce Vmax times S1. If I have a more complex chemical reaction, for example, quite often a reaction S1 goes to S2 actually has the Michaelis form, which is what we did earlier in class. Then the base reaction would be S1 over K Michaelis plus S1, saturated rate of production. But I still regulate in the same way by multiplying by K to the N over K to the N plus R to the N. So I just have a product of the reaction that would happen without regulation times the regulatory term. Now, is it always true that the rate of production by itself and the regulation are independent of each other, so I can just multiply them? Well, in complex reactions, no. The answer is it's not always true. But in most cases, it's pretty close. And if I don't know anything else, that's where you start in making your guess. And so if I have a metabolic network and I don't know anything except the arrows, these are the equations you start with. And the reality will be more complicated. But this will allow you to build a metabolic network just from the diagram that you can actually run and solve on a computer. And then you could begin to ask the question, gee, it doesn't quite fit the experimental data. How do I refine it? Because if you can't run it to begin with, you have no way of fitting anything. And so this allows you to take the diagrams you find in KEGG or in some other database and turn them into instantiable computable models with a first guess about how they're going to behave. And for a lot of purposes, the quality of this prediction is as good or better than the experimental measurements. Now, these days with human metabolism, people have made so many measurements in such accuracy. Well, or for E. coli, they've done it with such accuracy that that they can actually tell the difference. What is the value of what is the value of that exponential n? They can tell you that because they've made the measurements very, very accurately. But in general, until the past 5 years, we didn't know enough to be able to say better than this. This was about as good as you could do. And so there was no point in trying to build a more elaborate mathematics when the experimental data couldn't distinguish. And this is still the place to start when you're looking at a network. And so, at some level, if you get nothing else out of this course, this slide is the one thing I would like to walk away from, which is I look at a network and I say, what do I do with this? I don't know anything about the mathematics of this network. You're not helpless. You could take that network and you could make it a computable model, and that computable model will make sense, and it will give you meaningful answers. Will they be exactly what happens in the experiment? No. But will they be close enough that you could begin to refine things? Yes.
And also they eliminate completely the possibility of your dynamical system being
Nothing will diverge, nothing
Yeah,
will become
diverge.
unstable, everything will be well behaved.
Yeah.
And so then if you write your complicated network, you run it and it goes boom, you've made a mistake.
Yeah.
Because if you use this method, this is guaranteed to give you bounded soluble results. Now, the last thing that we're out of time, so I just show you quickly. If it's a gene regulatory network, basically everything carries through. P1 promotes the production of P2. That's interpreted as nothing goes to P2 at a rate that's promoted by P1. And it has exactly the same form. With gene regulatory networks, there are a couple of other things you might want to think about. One, with a gene regulatory network, you always have to remember that proteins degrade. So you always have to interpret that as P goes to nothing again. And also for genes, typically the gene is transcribed at what's called a basal rate. Even if you have no promoter, that polymerase will occasionally walk down that gene and make a little bit of messenger RNA. Now, if the gene is methylated, you won't get any of it. So it's not always the case that it's produced at a basal rate. And so the rate of production of the product then is our Hill function like that, plus B1, which is the basal rate of production. But when you see P1 promotes P2 at a gene regulatory network, you have an assumption of a Hill function for the production of P2 by P1, and you have to always remember you have to include the decay of P2, otherwise things will blow up. So for that, you have to add one extra factor, two extra factors, one, the basal rate of production, and two, the decay. Again, K is the half maximal concentration, N is the Hill exponent, B is the basal rate of production, and you have the Vmax. And so now you could do all of that, and I'm not going to go through that. You could look at the slides if you're interested, but you could go through the whole thing for inhibitors and activators, and that's your basic result. Typically, in gene regulatory networks, we assume that N is 4 or 8 because the turn on and turn off are fairly sharp. K in this case is not something you're necessarily going to keep constant for a gene regulatory network because K sets the concentration at which the gene turns on. If P1 is bigger than K, the gene is mostly on. If P1 is less than K, the gene is mostly turned off. So you're setting the threshold for turning the gene on.
You said B1 the same for every gene.
So Vmax is the rate of production.
Beats. Okay.
So the basal rate of transcription, that's something that we did a little bit of last week without having done this. So we played with that circuit without really describing what it does. In the homework, I'm going to ask you to do a couple of simple gene regulatory circuits. And I'll ask you to play with B and see what it does. Essentially, what you're going to find is if B is too big, effectively the gene is always turned on. If B is 0, then usually B is pretty small, and so it doesn't have a big effect. But it's almost impossible to, unless you actually do a knockout where you physically, do CRISPR and cut that gene out. If the DNA is there, there's always a little bit of leakage. And so it's important to say, if B isn't exactly 0, does it change anything? Most of the time the answer is no, but occasionally 0 and 0.01 really are different. And so it's important to check that. The other thing that'll happen is the following. Suppose that I have autoactivation the way we did last week. If I have autoactivation and I start with none of something, then my rate of production is 0. And so I just go 0, 0, 0, 0, 0. On the other hand, if I started with just a little bit of it, I could get a very different result. If I have a small basal rate, then that accident of having exactly 0 of the protein or the promoter doesn't matter anymore. And so it basically makes the system a little more tolerant to have the basal rate not be quite 0. I'll just show you this last slide because there was a question about it. Antimony, and you can do this as a homework exercise. Antimony does allow you to have variable stoichiometry. So if you wanted to change the exponent in your Hill function in antimony, you can do it. The key thing is you have to have a space. So up here, E plus N space S. N is the stoichiometric coefficient, and now you can scan N. And so why not try that out at home, see what it does. Most of the time, you're not going to scan N, but because if it's a real chemical reaction, then the stoichiometry is what it is, right? If you have H2O, the 2 isn't changing. It's really only for mathematical exploration. I hope that was fun for people. I realize that compared to some of the big ideas we were talking about earlier, these are very simple things, but I hope that this illustration of how to go from arrows to math, it was pretty fast. So I'd encourage you to look back at it. I don't know many textbooks which actually talk about that. And to me, it's actually one of the more important things to learn about how to work with these things. And I'm not really sure why that isn't emphasized in the textbooks, because that would be great. Okay, thanks everybody. Really appreciate you coming this week, and I hope we could have fun together on this.